In this series of lectures, we'll examine the endocrine system and its control of various functions in the human body. We'll be looking at the endocrine system as a foil to the nervous system, which we considered earlier in the course. And we'll be looking at comparisons between the two systems of control, both nervous and chemical. Instead of looking at each of the individual endocrine organs separately, we'll be considering them as a whole. Now, chemical signals in humans share a number of functions and a lot of overlap with the nervous system. So the first thing we're going to do is to compare the two systems of control, the means of control, and how they're similar and how they are different. So the human body basically has two systems of internal control, the nervous system, which we considered earlier, and also the endocrine control of the body, or hormones. Now, the images at the bottom of the slide really show the differences, many of the differences anatomically, between nervous control and chemical control of a situation. We saw with nervous system that there had to be direct contact between a nerve cell and its target cell, whatever that target cell might be. A nerve cell, as shown here, or a muscle cell, or a glandular cell, or another kind of cell, whatever the target cell might be. There had to be physical contact between the nerve cell and the target cell. Well, almost physical contact. There was, of course, a synapse and a synaptic cleft, or a space between one cell and the next. But there was direct connection. There was the communication of an electrical synapse, or an electrical depolarization, which may be transmitted via gap junctions, or there may be a neurotransmitter, which would be received and recognized by the target cell. But the direct physical connection between one and the other was key. Not so for the endocrine system, not so for chemical control of processes. In this case, we have a control system that acts at a distance. The endocrine cell, wherever it may be in the body, will secrete its substances, its very active substances, directly into a blood vessel or into the extracellular fluid that might bathe another cell. The target cell may be nearby, or it may be in the other a very distant portion of the body. And the communication is made not by direct contact, not by physical closeness, but by the delivery of a chemical messenger from one place in the body to another. So physically, these two systems differ greatly. However, there are a number of overlaps between these two systems. We saw with the nervous system that the means of communication was either electrical, by means of an action potential, or chemical, by means of a neurotransmitter, or even both. In the endocrine system, it is only a chemical communication, and it is delivered via the bloodstream or by extracellular fluid. The distribution of signal in the nervous system is restricted, one-to-one. One cell must communicate with the next. But we've seen in the endocrine system that a signal may be broadcast to the entire body if necessary. So the effect of the secretion of a hormone is widespread. The response to a nervous signal is a very quick one. We've seen, especially with saltatory conduction, that it is on the order of milliseconds from a stimulus to the end of the stimulus, and it may stop extremely quickly. However, with the endocrine system, the response may be very slow. It may take days to receive and respond to an endocrine message in another portion of the body. And the stimulus, even though there may be a cessation of the secretion of a hormone, the effect of that hormone may continue for a very long time as well. So some major differences between the two systems, but they have some things in common. One of the things that they do have in common is the type of cell that may either stimulate another cell or produce a hormone. We saw when we considered the renal system that there were specialized cells in the hypothalamus that secreted antidiuretic hormone. And these are neurosecretory cells. Neurosecretory cells can be seen in other portions of the body and certainly are cells that occupy both spaces. They can be nervous cells, and they can also be portions of the endocrine system. The endocrine system may also use some of the same chemicals, neurotransmitters, as the nervous system does. In other words, some neurotransmitters may also act as hormones, both local and far-reaching. And they may integrate with each other. In other words, there may be a stimulus, a nervous stimulus, that may stimulate an endocrine response. As a matter of fact, we saw that already in our consideration of the kidney and its regulation of blood pressure via antidiuretic hormone. There may be a stimulus from the central nervous system, the medulla, which will stimulate the release by a neurosecretory cell of ADH. So one system may activate another, and therefore they are very, very similar to each other. Now, hormones actually don't have a very long history in terms of our understanding of them. The very first human hormone was discovered just in 1905, and it's a hormone that we don't talk much about. It's called secretin, and secretin is a hormone that is a digestive hormone. It is secreted by a portion of the duodenum, and it was shown by the neurophysiologist, or I should say the anatomist and neurophysiologist Starling, to be a substance that was contained within fluid. It was dissolved within fluid. It was it was something that was contained within a fluid and was distributed by the circulatory system. So secretin, which will stimulate the secretion of buffers and other substances from the pancreas into the duodenum during digestion, was the first hormone discovered. So hormones have a number of similar characteristics. They're all fairly small molecules, and therefore they can easily be transported by the blood. They are extremely potent at very low concentrations. Some are even effective at a billionth molar concentration. So very, very potent stimulators of activity. They are secreted by glands. They're called endocrine glands because they do not have ducts, such as other glands, such as salivary glands, in which the secretion is directed towards a duct, and there's only one avenue for secretion. With endocrine glands, there are no ducts. These substances are secreted directly into the bloodstream. They're transported in blood, except for those hormones which are extremely local, which we call autocrines or paracrines, and they may be secreted directly into the extracellular fluid that bathes cells and tissues. They act on specific target cells. That's very similar to the nervous system, of course, but they must be recognized by receptors on the targets. Now, we've seen that in the nervous system that is true as well, that there are specific receptors that recognize and bind to neurotransmitters. The same is true of the target cells for hormones. And interestingly enough, hormones are very evolutionarily conservative. For example, insulin, a very small molecule, a very potent molecule, is the same in many mammals. Cows, chimps, humans, all make the same molecule and use it in the same fashion. There's also a molecule which acts as a stimulus to lactation in humans, prolactin. It also occurs in fish, same molecule, different function. So the molecule itself is evolutionarily conservative, if not the function of the molecule in the context of the human body. Now, hormones basically are of three chemical classes, and they are classified not only because of their structure, but also, very importantly, by their solubility in water or lipid. The first type is the peptide or the polypeptide hormone. And polypeptide hormones are very small chains of amino acids. They can be as few as 3 amino acids. They can be as many as about 200, but still they're extraordinarily small. We wouldn't call them proteins by any stretch of the imagination. And these are all water-soluble hormones. Next, there are amino acids, and many of these are derived from a single amino acid, tyrosine. These might include thyroid hormones and also the neurotransmitters, which are also hormones, epinephrine, norepinephrine, and dopamine, which are collectively termed catecholamines. Now, strangely enough, thyroid hormones are derived from tyrosine, and they are lipid-soluble, but the catecholamines are water-soluble. So even though they share the same source, they are not soluble in the same way. And last, there are steroid hormones, and the steroid hormones are fairly familiar to us. We've already discussed aldosterone, for example, made by the adrenal cortex, I should say medulla, no, cortex, I'm sorry, and also cortisol, and then the hormones estrogen, progesterone, testosterone, dihydrotestosterone, and all of these are derived from cholesterol, a neutral lipid, but many of these are not neutral lipids. They are all, however, lipid soluble. Now, why would we concern ourselves with whether the solubility of a potent substance was either in water or in lipid? Well, because there are very different effects of the hormones depending on their relative solubility in water or in lipid. Polypeptide hormones, as seen on the left and exemplified by the hormone secretin, which we just discussed a few minutes ago, must bind to receptors on the surface of a cell because, being water soluble, they cannot traverse the plasma membrane by themselves. So they must bind to a surface receptor, be recognized, the surface receptor must change its shape, and there are consequences in the cytoplasm of the target cell as a result of this binding. Amino acid derivatives derived from the amino acid tyrosine, as shown in the upper portion of this image here, usually bind to receptors on the surface of target cells as well, if they are not lipid soluble. And once again, it is the binding on the surface of the cell that will trigger the changes within the cell in response to the hormone. Now, lipid-soluble hormones, exemplified here by cortisol, are cholesterol-derived. Cholesterol is shown at the very top of the image. And they can enter the cell directly through the plasma membrane because they are soluble in lipid. And they will bind to a receptor in the interior of the cell once they have entered the cell. So this is the beginning of the mechanism of various types of hormones, and the mechanisms will differ, as we'll see in our next lecture.